All right, well, thank you very much for the opportunity to talk today about GB Science's plant-inspired Parkinson's drugs. I'm going to begin our talk today with a little bit of a corporate overview, and then I'm going to get right into the meat of the studies that we've done. So GB Sciences is a plant-inspired biotech developing prescription drugs. We have 5 preclinical programs in process, and we're preparing our lead candidate in Parkinson's for our first in human trial, and you'll learn more about that as my talk progresses. Our discovery program has yielded an intellectual property portfolio that covers over 60 serious health conditions. We have 6 US and 3 foreign patents that have been issued. We have 18 US and 49 foreign patents that are pending. And some of these patents cover our proprietary drug discovery platform, which is an AI-enabled drug discovery engine, wherein we're doing natural products research by combining traditional medical systems data with AI machine learning. And so our pipeline, as I mentioned before, and we're going to hear a lot more about our Parkinson's disease formulations, are in the late preclinical stage. The patent has been issued for this in the US, and we're aiming towards the clinic as soon as we can get there. We also have a chronic pain formulation that has the patent issued, and it's in the late preclinical stage. And we have both COVID anti-inflammatory and a heart failure programs. Those both, the patents have been issued, and we have animal proof of concept. We're also doing an anxiety formulation where the patents have been filed, and it's in preclinical studies right now at the National Research Council in Halifax, Nova Scotia, as well as some formulations for mast cell activation, wherein the patents have been approved or issued, and they're in early preclinical. So moving now into the main part of our talk, the identification of minimum essential therapeutic mixtures from cannabis plant extracts by screening in cell and animal models of Parkinson's disease. This work really wouldn't have been possible without the efforts of our collaborators, both at the National Research Council of Canada in Halifax, as well as both Chaminade University and the John A. Burns School of Medicine in Hawaii. In general, our GB Sciences drug discovery process starts with in silico work, wherein we're looking within natural products for putative candidates for drugs. And we then take the in silico generated leads and we take them through traditional cell and animal screening. What comes out the end are plant-inspired minimum essential mixtures. And we're creating these or producing these using synthetic copies of the plant-based active ingredients. And our intellectual property portfolio invokes both the synthetic and the natural versions of these mixtures. And now we really think that by aiming for these minimum essential mixtures, we're getting the best of both worlds. We have both the benefits of whole plant medicines, where we have thousands of years of use in these traditional medical systems. There are multiple active ingredients working in concert. These multiple low affinity ligands often have more than one disease-related target, but we really believe that that's the way that the human body works, is that things aren't usually simple. However, the plant sourcing can be varied, and it's difficult to achieve strict CGMP manufacturing standards if you're using whole plant medicine. Therefore, these whole plant medicines usually get relegated to nutraceutical products without US FDA or the equivalent pre-market safety and efficacy data. On the other hand, the traditional single active ingredient drugs, while they're very US FDA friendly and other regulatory agents, the single active ingredient view of the world sometimes is misleading. It would have you believe that every disease state can be reduced down to a single disease specific target, and the job then as drug discoverers is just to locate that one high affinity ligand that's going to make everything become realigned. The benefits of these single active ingredient drugs are also that the API manufacturing is usually very consistent. They're manufactured to strict CGMP standards, and the US FDA approval process and equivalence in international markets ensures pre-market review of safety and efficacy data. So as I mentioned before, what we're really trying to do in the process of trying to discover what minimum essential mixtures is to get both the best of both of these systems. We're trying to get the trying to capture the wisdom in these thousands of years of use in the traditional medical systems, also gain some gain of function and some synergy by having multiple ligands that can work together. But also we are looking to have the API manufacturing consistent and to have the safety and efficacy data that you would expect for any other prescription drug. So in the process of creating these minimum essential mixtures, we reduce the number of compounds through the sequential screening that we're going to talk about in a second. And then we get them down to 3 active pharmaceutical ingredient in each minimum essential mixture. These minimum essential mixtures also have the feature that we can demonstrate synergy in there, or synergy-like behavior, in that the molecule, if you take the effectiveness of the mixture, it outperforms the sum of the effects of the individual ingredients. And we are able to manufacture them under CGMP because we're using these synthetic APIs, which are homologs of the plant-based compounds. We are working very hard to understand the mechanisms of actions for these mixtures. We believe that having these mixtures there, we're really working with a more complete model of the biological targets that are regulating a biological disorder. Because the idea that one drug, one target is the only way to go, I think, is an outdated approach. So while it's more complicated, we do not believe that it's unknowable. We can figure out the mechanism of actions for these simple mixtures. We're combining these plant-inspired ingredients into proprietary oral delivery methods because we feel like this is a great way to increase the bioavailability and, of course, increase patient compliance. So for a further discussion of our Parkinson's minimum essential mixtures, I need to start with just a review of the basic concepts behind Parkinson's disease. So as we all know, Parkinson's disease is a multifactorial disorder. There is no one single cause of Parkinson's disease, and clinicians will tell you that there's usually a combination of inherited traits and environmental exposures that end up leading to the disease state. But at the cellular level, or at least, there are 3 different factors that are often considered. One is mitochondrial dysfunction. It's been shown that dysregulated calcium levels and a disruptive electron transport chain lead to the production of increased reactive oxygen species, which contributes to neuronal cell damage and death. Also, the alpha-synuclein protein misfolding and the formation of these alpha-synuclein fibrils inside Lewy bottles inside the neurons is the hallmark feature in the pathology of Parkinson's disease. And it's been demonstrated that these alpha-synuclein proteins also contribute to the damage and death of dopaminergic neurons. And then finally, the more recent discovery is the role of lysosomal dysfunction. Lysosomal dysfunction is related to the other 2 above, in that when the lysosomes are not working properly, you don't get the appropriate clearing of senescent mitochondria, and the lysosomes would also be what would clear misfolded proteins. And there's also some data suggesting that this lysosomal dysfunction interferes with the appropriate synaptic transfer of neurotransmitters, including dopamine release. At the system level, Parkinson's disease is also characterized by neuroinflammation, which contributes to the Parkinson's pathology, both in the brain and through neuropsychiatric non-motor, so both motor and non-motor symptoms, excuse me, as well as neuroinflammation has also been known to contribute to gut health issues in Parkinson's patients. Depending upon who you talk to, neuroinflammation might either be a cause or an effect of Parkinson's disease progression. And then Ultimately, the death of the dopaminergic neurons is another hallmark feature of the pathology, and it can be seen in the substantia nigra regions of the brain. Reduced dopamine levels are related to the Parkinson's symptoms. Those Parkinson's symptoms are broken down into both motor and non-motor symptoms. The three major motor symptoms, the three major categories of motor symptoms in Parkinson's include tremor, stiffness, and slowness of movement. The non-motor symptoms include, but are not limited to, difficulties with sleep, swallowing, digestion, and neuropsychiatric effects. Excuse me. So now that we've reviewed the basics of the factors involved in disease progression in Parkinson's, I'm going to now touch upon how, based on data that's already been released and published, how cannabis-based ingredients may in fact be used in Parkinson's therapeutics, and what might the mechanisms of actions be. So at the cellular level, we talked about the mitochondrial dysfunction and reactive oxygen damage. Multiple cannabinoids are known to be antioxidants in that they are scavengers of these reactive oxygen species. These reactions happen they're not dependent on any kind of receptor-based reaction. The molecules themselves are either electron receivers or donors in the neutralizing of these reactive oxygen species. It's also been shown that cannabinoids help mitochondrial dysfunction, oxidative stress, and neuroinflammation in different model systems. And significantly, it's been shown that CBD is able to restore calcium regulation in mitochondria that have been treated with MPTP, which is one of the neurotoxins that is used to mimic a Parkinson's disease type state. Another study has actually implicated TRP channels in this same, the TRP channel activation can actually help to restore the calcium dysregulation in the mitochondria. And we believe that these mechanisms are central to the ways that our mixtures are performing. At the systems level, there's also reports of Parkinson's involving both a desensitization of dopamine release. So it's not just that dopamine levels are going down, but there's also an uncoupling of the neurotransmitter release. And it's been shown that cannabinoids can help alleviate this desensitization of the dopaminergic system in Parkinson's disease. And that is not such a surprise when you consider that the major receptors for cannabinoids, CB1, CB2, and TRPV1 levels are incredibly high in basal ganglia, globus pallidus, and substantia nigra, which are the regions which will control movement. And the endocannabinoid system is known to neuromodulate dopaminergic, glutamatergic, and GABAergic systems. Specifically, the endocannabinoid system is involved in retrograde signaling at the synapse for inhibitory regulation of neurotransmitter release. And at the level, so basically, it's been shown that cannabinoids can shut down the release of the neurotransmitters, which is very relevant for the Parkinson's disease progression. At the systems level, we also find the death of the dopaminergic neurons to be significant, and we're using a OHGA-based model, and we've demonstrated the death, and others have demonstrated the death of the dopaminergic neurons in the substantia nigra using these compounds, and others have shown that cannabinoids can be neuroprotectants as antioxidants, calcium regulators, and for their anti-inflammatory processes. And then finally, we'll be looking at the ability for these cannabis-based ingredients to work through cannabinoid receptors in the basal ganglia regions to affect slowness of movement, tremor, and rigidity Others have looked at the slowness of movement, but we've got new data to support the idea that they can also affect tremor and rigidity. So, big picture of our screen. We started with metabolomic profiles from cannabis chemovars. And we did an in silico analysis of these profiles along with a literature review. And this was used to select the potential compounds and mixtures for study in the these cell-based areas. Because we're talking about there was potentially greater than 100,000 potential mixtures. One of the strategies we've used in our process is that we're not saying that we're going to test every single possibility, but we're using these different screens to pick the best candidate at each level. And the best candidate from each level gets brought down to the next level. So while we started off with this over 100,000 potential mixtures, when we got down to the complex mixtures, we tested over 1,000 potential mixtures, and we took the top performing mixture from the cell screens into the minimum essential mixture study where we were using the zebrafish and the OHGA. And now we have 3 drug candidates which are in rodent studies and also are being produced as oral dissolving tablets in preparation for a first in man. So in doing these screens, picking the screen is super important for both making sure we're going to get a potentially effective mixture, but also for understanding mechanisms of action. So at the cell level, we were looking at the mitochondrial effects using MPTP, which is a compound that was originally discovered as a byproduct when things went wrong, when people were making illicit Demerol on the street, and they found people that were this MPT, which is taken into cells through dopamine-specific transport systems, and then intracellularly, MPTP is cleaved by MAO, monoamine oxidases, to MPP, which poisons the mitochondrial electron transport train by dysregulating calcium. And so this is the assay that we used for all of our mixtures. We also use a dopamine secretion because we were interested in the idea that cannabinoids could also be used to repair the impaired dopamine release systems. So there we measured the percent increase in dopamine secretion relative to control. And so what we found is that at the level when we were looking at complex mixtures, and these are mixtures that contain 9 to 12 different ingredients. If you looked at the individual ingredients, you'll find that while there were significant effects of the individual ingredients tested singularly, the mixtures often outperformed that of the individual ingredients. And then one of our really surprises, we really thought that so these mixtures are the minor cannabinoids and terpenes that were in the let's say prodrugs. And what we found is that without the major cannabinoid, there still was a lot of activity, which was kind of a surprise for us. We assumed that we would take this addition of the major cannabinoids for there to be significant activity, and that wasn't the case. So based on this initial work, as I mentioned before, we started with the in silico screening of the 11 to 19 terpenes. We determined our desired profiles. We did our complex mixture screening. We rank ordered those complex mixtures. We chose mixture 1 because it was the best performing complex mixture for further study, because we needed to be able to reduce this 10 ingredient mix down to 3 to be able to have a minimum essential mixture. So mix 1 contained 5 cannabinoids, 2 major, CBD and or CBN, plus 3 minor cannabinoids, CBC, CBG, CBDV, and 5 terpenes, D-limonene, D-linalool, T-neurolidol, alpha-pinene, and phytol A. And so we started with a dose response of the individual cannabinoids. We did a 5 log scale dose response, and you'll see that all of the cannabinoids that were tested showed some neuroprotection against the MPTP in these dose responses. In contrast, if you look at the dose response of the individual terpenes, only the highest level of transneurolidol showed any protective effect when they were measured singly. So then we were asking, all right, well, which contributes more to our complex mixture one? Is it the cannabinoids or is it the terpenes? So as I mentioned before, mixture one contains potentially 10 ingredients, but these first two are the major cannabinoids that we're going to add in after. So mix one really contains the 3 minor cannabinoids plus the terpenes. Oops, back. And mixture two is all the terpenes tested together, and mixture three is the minor cannabinoids tested together. And then we looked at the effects of the mixtures, effectiveness of the mixtures, both by themselves or with the addition of the major cannabinoids. So what we determined is that the cannabinoids contributed to the mix greater than that of the terpenes. It's not to say that the terpenes didn't have some contribution to the effectiveness of the original mixture, but when we had to make a decision about whether we were going to focus on cannabinoids or terpenes or some of both, we decided to go ahead with looking at the mixtures of cannabinoids in the next level of analysis. So the next level of analysis is the zebrafish assays, where we were looking at the level of motor symptoms. We were using 6-hydroxydopamine, which is a neurotoxic synthetic dopamine that selectively causes death in dopaminergic neurons. And then what we were measuring was slowness of movement, or total distance traveled, which is the standard movement assay that's used for OHGA. In the fish, we also added an additional analysis, which is we were looking at tremor, which is a novel phenotype. And we were using pixel-wise movements, and we'll talk about that again a little bit later. So, as mentioned before, this is just proof that the 6-OHGA does in fact target the dopamine production. So, in the midbrain, which is in the circle, you'll notice that if you treat the animals with anti-tyrosine hydroxylase, which is a dopamine-linked biomarker, you get a good expression in this hindbrain. However, with exposure to OHGA, that signal just gets depleted over 72 hours. And we found a similar pattern if we were using cell death markers. For the movement studies, as I mentioned before, there's two kinds. If we're talking about total distance traveled, then we're looking at the center point analysis, meaning that the computer program is tracking the distance that this one center moves throughout the well over the time period where they're being measured. Whereas when we're looking at these activity measurements, which what we used to quantify the tremor type phenotype that we noted, they were looking at the percent pixel change in measurements, and this gives us a better idea of the quality of the movement, not just the total distance traveled. So, as you would expect, when you expose the fish to OHGA at 3 different concentrations, there was in fact a dose-dependent decrease in the total distance traveled, as had been reported by others. What was novel in our study is that we were also able to show that the average frequency of activity state switching, meaning going from twitching to not or to fight or flight, there was a statistically significant difference between the MEAOH, which is the vehicle control, and the OHGA, or the Parkinsonian-like state. In addition, we also showed that there was a statistically significant difference if you look at the cumulative duration in the activity, wherein the OHGA had spent significantly longer in the tremor-like state than the controls. So we then started to expose, after we had established the basic parameters for the OHGA model in those zebrafish, we then started to, so these fish were being exposed to 72 hours of OHGA, so that they're 2 days after they're hatched. They're exposed to the OHGA for 3 days, and 24 hours after the exposure to the OHGA, we started to expose them for 48 hours to the drugs, meaning these different minimum essential mixtures. What we found is that when we started by using equimolar MEM, meaning for the three, for example, for MEM-A, for example, it would be an equimolar ratio of CBC, CBD, and CBDV. We found that 3 of these that we tried did reduce the Parkinson's-like symptoms statistically, significantly. And so MEM versus control, and here we have the MEM versus the MEM plus the OHGA versus the OHGA. And so here we see these 3 are statistically significant. So we went further in our studies, and here we show the results of these same 3 equimolar MEM, but this time we're, instead of just looking at the total distance traveled like before, although the total distance traveled is represented here, and at 3 different concentrations. Here we're looking at the activity measurements, and you'll see very significant differences in the activity measurements at these different concentrations. So after the early work of looking at the equimolar ratios, we said, can we improve upon the performance if we alter the ratios? And so the way to read this table, each of the white squares represents the test results for a newly formed MEM, and that the ingredients in that MEM are indicated based on the first compound and concentration is the yellow bar at the top. The second one is the green bar that's directly above the white box, and then the third compound is the purple cannabinoid and concentration located to the left of that white box. And so if the box contains asterisks, then that means that there was a statistically significant reduction in the Parkinson's-like symptoms. If it has a 0, that means that there was no reduction. And if there was an A, it was meant that the early precursors of these particular ratios were not one of the best performers, so they were not continued in that particular line. And so after doing this whole matrix of the different ratios in the zebrafish, we found that 22 cannabinoid ratioed optimized MEM reduced Parkinson's motor symptoms. And then if we then took the top 6 and looked more closely at that, and these ones were what we considered these ones were all better than the original equimolar. They performed better than the original equimolar MEM. And so, in conclusion, in a zebrafish model of Parkinson's disease with 72 hours of OHGA exposure, we were able to restore overall movement levels using our MEM, which is based on the total distance moved. We also were able to reduce the resting tremor, measured based on frequency and duration and shifts in activity states. And we were able to maintain a normal startle response. There was statistically significant reductions of Parkinson's-like motor symptoms in 3 out of 10 of the equimolar MEM, in 22 out of 63 of the cannabinoid ratio controlled MEM. 5 outperformed the other MEM. 3 have now been selected for formulation and ADMET testing as clinical trial prototypes. And these results suggest some things about the mechanism of actions of our MEM. At the cell level, our MTP-TP assays suggest that cannabinoids were able to protect against MPTP, and the mixtures enhance the protection against the MPTP. Also, in the dopamine release assays, cannabinoids increase dopamine release, and the mixtures enhance this dopamine release. At the system level, the anti-TH visualization in the midbrain of zebrafish confirms death of the dopaminergic neurons there, and the cannabinoids appear to be neuroprotectants. Also at the system level, in the 6-OHGA-induced Parkinson's motor symptoms, mixtures improve the total distance traveled, and mixtures can increase the on periods with no tremor. In summary, our plant-inspired strategy is based on looking for minimum essential mixtures, where the whole plant efficacy is retained, yet the manufacturing efficiencies of single ingredients can also be attained. Our MEM retain the molecular synergies of the complex plant extracts, but they're simplified to 3 compounds. For our active ingredients, we're using synthetic homologs of plant compounds because the synthetic homologs are identical in structure to plant compounds and with regulatory and supply chain advantages. And they can be manufactured under CGMP. The delivery route that we're using is an oral route. And for our Parkinson's tablets, and for our Parkinson's formulations, we're using the Zydis orally disintegrating tablets from Catalent Pharma. These unique freeze-dried oral solid dosages offer instant oral dispersion, typically less than 3 seconds. And for our Parkinson's MEM in the Zydis, it's a convenient dosing solution for Parkinson's patients, especially given that greater than 50% of Parkinson's patients have swallowing problem, has particular clinical advantages. And this research wouldn't be possible without the help of our collaborators at Chaminade, Michigan State, the University of Hawaii, the University of Sevilla in Spain, the University of Cadiz in Spain, the NRC in Canada, the University of Lethbridge in Canada, the National and the University of Athens in Greece, as well as our development partners, Pyrasis, who supplies the homologs of the plant compounds, and Catalent Pharma. And thank you to our scientific advisory board. Here are some references, and that's it.
Thank you for your presentation. Now floor is open for the questions, if anyone have.